In this next lecture series, we're going to briefly review Mendelian genetics, and in particular, the laws of inheritance. Now, you've probably all heard already of Gregor Mendel. And in essence, in this particular slide, we're going to cover the two theories that started out. The first was the blending model of inheritance versus the particulate model of inheritance. And now, before Mendel's experiments, prevailing thought was that heritable characteristics came from the idea that contributions from each parent somehow blended together, hence the blending model of inheritance, to generate the inheritance patterns in the offspring. So, for example, if you had a very tall father and a very short mother, they would ultimately produce children of intermediate height. Now, I will put in a quick digression here that actually human height is determined by many genetic and environmental factors. But in essence, then, this blend would then pass on to the next generation. Now, the problem with this model, though, was that it allowed for no variation over time. And in essence, in our particular example, ultimately, everyone then would be of average height or average size or uniform in height. And clearly this is not what happens. So it became somewhat clear that maybe this model for inheritance was not correct. Now, Gregor Mendel came along, and we'll talk about his experiments in a little bit more detail. But according to Mendel's particulate model, as an alternative to the blending model, an individual receives some form of a particle from each parent. Now, we know today that this particle is called an allele, and ultimately it's a chromosome coming from each parent. And those chromosomes contain genes, and the genes from each parent are known as alleles. And alleles represent the many forms that a gene can take. And then it's ultimately these two particles, or two alleles, that dictate the inheritance pattern in the offspring. Now, returning to the example we used previously, if you have a very tall father and a short mother, they could still produce offspring of average height. However, in the particulate model now, the contributions from the parents are separated from one another when being passed on to the next generation. Thus, in this way, the particles can continue to combine in various ways, and thus you see variation being maintained over time. And so let's talk about how Mendel ultimately then came to this particulate idea of inheritance. And so the model that Mendel used, as you may already know, are these pea plants. Mendel found that pea plants were very easy to breed. They had distinctive visual traits or phenotypes, and that's what you can see, the phenotype. And examples of different phenotypes in pea plants include the plant height, the pea color, the flower color, and the texture. And so what he did is he first established pure breeding lines for each of these traits. And he would call these the pea or the parental lines. And when two of the same line were mated, the offspring always were identical to the parents. And that's what it means to be a pure breeding line. And then next he selected for a particular characteristic, such as plant height. And then he mated two parents that were purebred for contrasting traits. So, for example, once he had a pure breeding line of tall plants and a pure breeding line of short plants, he would then mate a tall plant with a short plant. And what he found was that all offspring looked like one of the parental lines. In this particular case, all of the plants were tall. He then crossed these offspring, and these are known as the F1 generation. And what he found is when he crossed the F1 generation plants to produce an F2 generation, he had a combination now of 3/4 of the phenotypes were tall, as was found in the previous generation, and then the remaining 1/4 of the progeny looked like the other parental phenotype, and they were short. So what I mean by that is when he had crossed the two F1 generations, what he would find was 3 out of the 4 plants would be tall and 1 out of the 4 plants would be short. And so what this was beginning to tell him was that things were not blending together. He was, in essence, what he was seeing was this kind of variation, even when you bred plants that looked to be all tall or all short. And so there is sort of a trick that you can use. And how do we figure out what Mendel was determining with his pea plants? And one way of doing that, or working out Mendelian genetics, is to use something called a Punnett square. And it's simply this box here in the lower right-hand corner. And the Punnett square can be thought of as a bookkeeping tool. It allows us to work out all the different possible combinations of male or female gametes for a particular cross. So for the sake of simplicity, we will refer to these male and female gametes as sperm and egg. The specific form of the particle that is passed on to the offspring is called the allele. And so the boxes in the Punnett square help us to keep track of all possible offspring that can be produced by this particular cross. And so, as with Mendel's experiments on tall versus short pea plants, let's start with that same scenario and with this characteristic of plant height. So then each individual has two alleles that correspond to height, one allele inherited from each parent. And in this case, an uppercase T is going to represent the allele that confers tallness, and a lowercase t is going to represent an allele that's considered short, opposite of tall, if you will. Now recall that Mendel started with pure breeding lines of tall and short plants. Thus all individuals of our tall line have only T alleles, and all of the individuals of our short line have only lowercase t alleles. The allelic combinations or genotypes of the tall individuals are uppercase T, and the allelic combinations or genotypes of the short individuals are lowercase t. Individuals or genotypes that have two copies of the same allele are called homozygotes. And so in order to be a homozygote, what you're going to see is the two capital letters together or the two lowercase letters together. So in this demonstration, we're going to cross a tall male with a short female. We are going to get the same results if we crossed a tall female with a short male. However, in more complicated crosses, the direction of the cross with regardless to sex can make a difference. We will not worry about that at this time. During fertilization, each parent contributes only one allele for plant height, even though each parent has two alleles. So in our example, both alleles from the male parent are the same, written capital T, capital T. Similarly, both alleles that could be contributed by the female are the same, right? Both lowercase t. So here we write the potential combinations on the top and potential female contributions on the left. And by putting one T in each sperm, the alleles are following Mendel's first law of inheritance called the law of segregation. The two alleles that made the male tall are now segregating to produce reproductive gametes that can combine with contributions from the female to form the next generation. And the same is true for the females. And so you see that here. You see the males, first each male passes on his alleles, one per gamete, to the reproductive cell. And then following that, what you see, then the female is going to pass on one allele, one per egg. And so crossing these two parents, these two pure breeding parents, tall males and short females, ultimately results in offspring that have this genetic combination, uppercase T, lowercase t. Now these are considered heterozygotes because a genotype with two different alleles, an uppercase letter and a lowercase letter, is known as a heterozygote. In pea plants, the capital T allele masks the effects of the lowercase t allele. And so in essence, the terms dominant and recessive are used for the masking and the covered allele, respectively. So what we say is that this is a heterozygous genotype, yet all offspring from this cross are tall for their phenotype. And again, the phenotype is the observable feature produced by the genotype. So these alleles thus follow Mendel's third law of inheritance, the law of dominance. And so where we have an uppercase letter that usually denotes the dominant allele, the lowercase letter denotes the recessive allele. Unlike human height, which is determined by multiple factors, plant height is determined by only one major heritable factor. And in addition, dominant alleles do not always cover up the recessive allele entirely. Sometimes the dominant allele only partially masks the recessive allele in a process called incomplete dominance, but we will not go into any more detail about that. Now, if we have these heterozygotes in this F1 population, and we now cross the two heterozygotes, you see them here. This was the second part of the experiment that Mendel did. What you get then, and if you just want to determine what the possible genotypes are, you write the allelic combinations on the top and the side of the Punnett square. And now first write the two possible alleles that the male contributes to the Punnett square. And so the male can contribute either an uppercase T, or the male can contribute either a lowercase t, and then continue with the contributions that the female can give. You see the female can contribute either the uppercase T or the lowercase t. And what you find here now is a different combination. And so when the F1 generations are crossed, there are three possible genotypes in the ratio of 1 to 2 to 1 for the uppercase T, uppercase T, uppercase T, lowercase t, and lowercase t combinations. Now, since the uppercase T allele is dominant to the lowercase t allele, every individual with at least one uppercase T is going to be tall. So in this case, you see this individual, and this individual would all be tall, and only those individuals with the genotype of a lowercase t would be short. So while we find a genomic or a genotype ratio of 1 to 2 to 1, what we find is a phenotype ratio, and this is what we see in the individual, of 3 to 1 for tall to short respectively. Notice then how this particulate model of inheritance is maintaining variation. Under the blending model of inheritance, we would expect a mating of two tall individuals to produce only tall offspring. But now instead, what we have are two tall offspring, and the crossing of the F1 generation, we have recovered the variation that we saw in the parental line, and yet again the appearance of a short plant. Just to expand on this so you're aware, Punnett squares can be expanded, and you can actually consider two traits at a time in this particular Punnett square. The two traits being looked at are again tall, capital T for the tall dominant gene, lowercase t for the recessive short height in plants. And then you can also add on, in this case, they did an uppercase R and a lowercase r for round and wrinkled seeds respectively. And so if you expand out the Punnett square, much like you did looking at the different allelic combinations in the single test crosses, you can also build Punnett squares for double test crosses. We will not do double test cross Punnett squares on an exam, but just to make you aware of how the system can work, so you can again use the same process in essence to look at different allelic combinations and to be able to determine particular phenotypes and genotypes of an individual. It gets much more complicated when you move from the single to the double test cross situation. I left these here, though, in case you're interested in studying how the different combinations occur, but you're not responsible for doing these kinds of crosses on your own.